Hello students, welcome to EPG Padasala. I am Dr. P. Nagaraj from Anna University, Chennai, working as assistant professor. Today we are going to talk on module A, carbohydrates interaction with other biological molecules from the paper of structure and function of biomolecules. Objective of today's lecture is role and significance of carbohydrates, reactions of monosaccharides and oligosaccharides, polysaccharides. Of course, glycoconjugates also going to study in details. Carbohydrates, protein interactions also going to be studied in details. So, role of carbohydrates as a major energy source for living organism, carbohydrates. Glucose is the principal energy source in animal and plants as means of transporting agents, means it is act as a transporting agents, like example sucrose in plant tissues. As a structural material, most of the carbohydrate act as a structural material. Examples cellulose in plants, chitin in insects, building blocks of nucleosides, etc. As a precursor for other biomolecules like a purine, pyrimidine that can be obtained from carbohydrates. Significance of carbohydrates, again, carbohydrates are the most abundant biomolecules in nature, having a direct link between solar energy and the chemical bond energy in living organism. Example photosynthesis, which is happening using direct sunlight, CO2 and water, it will produce a carbohydrate or polysaccharide via photosynthesis. And another thing is the source of rapid energy productions, easily you will get energy, and structural building blocks of cells, cell walls, everything made up of carbohydrate unit. Apart from that, components of several metabolic pathways, whatever metabolic pathway which undergo several pathway, and recognition of cellular phenomena such as cell recognitions and bindings by other cells or hormones or viruses, which is the significance of carbohydrates. Examples of carbohydrates, which is glyceraldehyde, which is the simplest form, then that hexose is D-glucose, again ketose is D-fructose, which will come pentose sugar is ribose, and will also give sucrose. These are all the examples. So properties and classification of carbohydrates, again the typical properties are, which is a colorless, crystalline solids, soluble in water but insoluble in nonpolar solvents, and one of the carbon atom is double bond to an oxygen atom to form a carbonyl group. Each of the other carbon atoms has a hydroxyl groups. Carbohydrates with an aldehyde functional group are called aldoses. Example glyceraldehyde. Those with a keto group are known as ketosis, dihydroxyacetones. And this classified according to the number of carbon atoms they contains. Classification of carbohydrates, again further, which is monosaccharide, aldoses, ketoses. Examples, if the aldehyde group present in the molecule, which is known as aldoses, if keto group present in the molecule, which is known as ketoses. So if it is a trisaccharide, means only 3 carbon which is present, which is called triose. If 4 carbon present, which is a tetrose. If 5 carbon present in the molecule, which is called pentose. If 6 carbon available, that is called hexose. If it is aldehyde, aldohexose. If it is keto group, then ketohexose. Monosaccharides, stereoisomers. Isomers, same chemical formula, different in structure. Conformation in case of the spatial arrangement of substituent groups, which is vary, will give conformation. Chiral centers, asymmetric carbon, that is carbon atom with 4 different substituent will produce chiral center. Enantiomers are mirror image of stereoisomer, and the simplest aldose glyceraldehyde contains 1 chiral center, the middle carbon atom, that has 2 different optical isomers are enantiomers, which represents simplest chiral carbon. Cyclic structure of monosaccharide. So the monosaccharide also have open chain form or cyclic form, 2 form of structural information available. In case of aqueous solution, the monosaccharides with 5 or more carbon atoms in the backbone occur predominantly as cyclic or ring structure in which the carbonyl group has formed a covalently bond with the oxygen of hydroxy group along the chain. So it will form either 5-membered ring or 6-membered ring depend upon the flexibility or number of hydroxy group available in the carbon chain. Pyrenosis and furanosis. In case of pyrenosis, 6-membered ring compounds resembles pyren means 6-membered ring, whereas furanosis is a 5-membered rings which is resemble furan. The structure systematic names glucose fructose becomes shown in the picture alpha glucopyranose and alpha D fructofuranose. Similarly, beta D glucopyranose, beta D fructofuranose. The furanose indicates 5-membered ring, whereas pyranose indicates 6-membered ring. Fischer and Haworth form of sugars also known. This is another way to represent the molecule, where the straight chain molecule having the aldehyde group and the C1 carbon, which is having hydroxy group C2, C3, C4, C5 and in fact C6 also. So the linkage between these two via C5 carbon and C1 carbon will make 6-membered ring. If the hydroxy group above the plane, which is beta, if it is below the plane, that is alpha, or also axial hydroxy group will give alpha or equatorial hydroxy group will give beta form. Important reactions monosaccharides are mutarotations, which is well known that alpha sugar will convert to beta sugar, and the second one is oxidation. The hydroxy group will undergo oxidation, will produce aldehyde or ketones, and the third one is a reduction. The aldehyde group which will undergo reduction to produce alcohol, and the fourth one is isomerization. Isomerization is one form of sugar unit converted to other form. Esterification, the hydroxy group easily converted to ester using acids. Similarly, glycoside formation, where the ether linkage will form via combining 2 monomers or 2 moles of glucose unit will give glycosidic linkage. Important reactions of monosaccharides, which is first one is it will undergo mutarotations, and second one is oxidation and reductions, third one is reductions. So in case of mutarotation, alpha and beta forms of sugar are readily interconverted when dissolved in water. So alpha will convert to beta, beta will convert to alpha, and the same way oxidation reactions in presence of oxidizing agent, metal ions like a copper 2 plus and enzymes, monosaccharides undergo several oxidation reactions. The oxidation of aldehyde groups yield aldonic acids, of terminal CH2OH yield uronic acids, and both the aldehydes and CH2OH gives aldaric acids. The carbonyl group in both aldonic and uronic can react with the hydroxy group of the same. So this is what in case of oxidation reaction, whereas in case of reduction, aldehyde or ketone groups monosaccharide yield sugar alcohol, namely alditols. Sugar alcohols, example sorbitols, are used in commercially in processing foods and pharmaceuticals. Example molecule to form a cyclic ester lactones. Another class of reaction which undergo isomerizations, esterifications and glycoside formations. Isomerization nothing but monosaccharides undergo several types of isomerizations. Example, D-glucose is an alkaline solution several hours containing D-mannose and D-fructose that will undergo isomerization. In case of esterification reactions, the free hydroxy group of carbohydrates react with acids to form esters. This reaction changes the physical and chemical properties of sugar, and also glycoside formations, hemiacetals and chemiketals reacting with alcohol to form the corresponding acetal or ketal. On the contrary, when a cyclic hemiacetal or hemiketal form monosaccharide react with alcohol, the new linkage called glycosidic linkage and the compound is called glycosides. Reducing sugars. All monosaccharides are reducing sugars. They can be oxidized by weak oxidizing agents such as Benedict reagents. Benetic reagent is solution of copper sulfate, sodium hydroxide and tartaric acids. Aqueous glucose is mixed with the Benetic reagents and heated. The reaction reduces the blue copper 2 ion to form a brick red precipitate copper 1 oxide. Because of this, glucose is classified as reducing sugar. Diabetes mellitus characterized by high blood glucose level that spills over into the urine. This high glucose level impairs the insulin stimulated glucose entry into cells and starve the cells of insulin. This leads to ketosis or high level of ketone bodies that hinders the buffering capacity of the blood in the kidney, which controls blood pH. The H plus excretions is accompanied by excretion ammonia, sodium, potassium and phosphate ions, causing severe dehydrations. This leads to excessive thirst, symptoms of diabetes, of life-threatening decrease in blood volume. Importance of monosaccharides are glucose, fructose, galactose. In case of glucose, D-glucose is the primary fuel in living cells, especially in brain cells that have few or no mitochondria. Cells such as eyeballs have limited oxygen supply and use large amount of glucose to generate energy. Dietary sources includes plant starch and the disaccharides lactose, maltose and sucrose. Importance of monosaccharides are glucose, fructose, galactose. In case of glucose, D-glucose is the primary fuel in living cells, especially in brain cells that have few or no mitochondria. Cells such as eyeballs have limited oxygen supply and use large amount of glucose to generate energy. Dietary sources includes plant starch and the disaccharides lactose, maltose and sucrose. Important monosaccharides in case of fructose, D-fructose, also known as levulose, is often referred as fruit sugars and is found in some vegetables and honey. This molecule is an important member of ketose member of sugar. It is twice as sweet as sucrose per gram basis and is used as a sweetening agent in processed food product. It is present in large amounts in male reproductive tract and it is synthesized in the seminal vessels. Another important monosaccharide, galactose, is necessary to synthesize a variety of biomolecules, lactose in mammillary glands, glycolipids, certain phospholipids, proteoglycans and glycoproteins. Galactose and glucose are epimers at C4 carbon and interconversions is catalyzed by enzyme epimerase. Medical problems, galactosemia, genetic disorder where enzymes to metabolize galactose is missing. Accumulation of galactose in the body can cause liver damage, cataracts and severe mental retardations. Monosaccharides derivatives, again uronic acids formed when terminal CH2O group of monosugar is oxidized. Important acids in animals, D-glucuronic acids and its epimer L-ioduronic acids. In liver cells, glucuronic acids combines with steroids, certain drugs and bilirubins to improve water solubility therapy, helping the removal of waste products from the body. These acids are abundant in the connective tissues, carbohydrate components. Aminosugars is a derivative of monosaccharides. Sugars in which hydroxy group, normally second carbon, is replaced by amino group, example D-glucosamines and D-galactosamines. The common constituent of complex carbohydrate molecules found attached to cellular proteins and lipids. Amino acids are often acetylated, example N-acetyl glucosamines. This also play major role. Again, deoxysugars present in the monosugars. Monosaccharides in which an hydrogen has replaced an OH group. Important sugars L-fucose and 2-deoxy-D-ribose. L-fucose found among carbohydrate component of glycoproteins such as those of ABO blood group determines on the surface of red blood cells. 2-deoxyribose is the pentose sugar component of DNA. Glycosides, again monosaccharides, disaccharides, oligosaccharides and polysaccharides. It can be linked by glycosidic bonds, and more than 12 monosaccharides units present in polysaccharides. Example, glycogens, homopolysaccharides having hundreds of sugar units. Glycosaminoglycans, heteropolysaccharides containing a number of different monosaccharide species, which is examples for glycosides. The disaccharides and oligosaccharides, again configuration is alpha beta. Digestion aided by enzymes. Deficiency of any of the enzyme causes unpleasant symptoms. Fermentation in colon produces gas. Most common deficiency in menstrual disorder. Lactose intolerance caused by reduced synthesis of lactase, etc. are present in the problem of disaccharides. Importance of disaccharide, especially lactose. Disaccharide found in milk composed of one molecule of galactose and glucose linked through beta-1,4 glycosidic linkage. Because of the chemical hemiacetyl group of the glucose component, lactose is a reducing sugar. Why? Because the one sugar unit having C1 hydroxy group free, it can undergo rearrange the open the ring and it may form aldehyde group, which is responsible for reducing sugar. Lactose intolerance, where lactose in an infant is hydrolyzed by intestinal enzymes lactase to its component monosaccharides for absorption into the bloodstreams. Most adult mammals have low level of beta-galactosidase, hence much of the lactose they ingest move to the colon, where by bacterial fermentation generate larger quantities of CO2, H2 and irritating organic acids. These products causes painful digestive upset known as lactose intolerance and is common in the African and Asian descent. Maltose is another class of disaccharides, an intermediate product of starch hydrolysis. It is disaccharide with an alpha-1,4 glycosidic linkage between two glucose molecules. In solution, the free anomeric carbon undergoes muta-rotations, resulting in an equilibrium mixture of alpha and beta maltoses. It doesn't occur freely in nature. Sucrose is another class of disaccharides, common table sugar, cane sugar or beet sugar, produced in the leaves of or stems of plants. It is a disaccharide containing both alpha glucose and beta fructose residues linked by alpha beta 1 to glycosidic bond, which is responsible for the disaccharides. Again, cellobiose, degradation product of cellulose containing two molecules of glucose linked by a beta 1,4 glycosidic bond, doesn't occur freely in nature. Oligosaccharide sugars. Oligosaccharides are small polymers often found attached to polypeptides in glycoproteins and some glycolipids. They are attached to membrane and secreted proteins found in endoplasmic reticulum and Golgi complex of various cells. Two classes, N-linked glycopeptide and O-linked glycopeptides. Polysaccharide composed of large number of monosaccharide units connected by glycosidic linkage, which is again classified on the basis of their main monosaccharide compound and the sequence and its linkage between them, as well as the anomeric configuration of linkage, the ring size and absolute configuration DRL and other substituent present, etc. Polysaccharides are more hydrophobic if they have a greater number of internal hydrogen bonds, and as the hydrophobicity increases, there is a less direct interaction with water. So, which is divided into homopolysaccharides and heteropolysaccharides. We already discussed in details. Classification of polysaccharide. which is classified homopolysaccharides, heteropolysaccharide in broadly. Homopolysaccharide again, which is further classified unbranched, branched two way, whereas in case of heteropolysaccharides, two monomer types of unbranched and multiple monomer types branched. So that means if you hydrolyze, you will get a single monomer unit, that is homopolysaccharide. If you hydrolysis, you will get different type of monomers, that is heteropolysaccharides. In case of homosaccharides, found in abundance in nature, which is naturally available, and important example which belongs to homosaccharides are starch, glycogen, cellulose and chitin. Starch, glycogen and cellulose all yield D-glucose when they are hydrolyzed. Cellulose, primary component of plant cells. Chitin, principal structure of component of exoceletrons of arthropods and cell walls of many fungi, yield glucose derivative of N-acetylglucosamine when it is hydrolyzed. And starch again, which is naturally abundant nutrient carbohydrate, is a polymer found in cheaply in the seeds, fruits, tubers, roots, stems, pith of plants, notably in corn, potatoes, wheat and rice, and varying widely appearance according to source, but commonly prepared as white amorphous tasteless powder. Any various substances such as natural starch used to siphon cloth as in laundering. Two polysaccharides occurs together in starch. One is amylose and amylopectin, which we studied early, and that we can see amylose is an unbranched chain, that means there is no branching. D-glucose residues linked with alpha-1,4 glycosidic linkage. In case of amylopectin, a branched polymer containing both alpha-1,4 linkage as well as alpha-1,6 glycosidic linkage, which is present in amylopectin. Starch digestion begins in the mouth. Alpha amylase in the saliva initiates hydrolysis of the glycosidic linkage. It's a class of homopolysaccharides. Glycogen is the storage form of glucose in animals and humans, which is analogous to the starch in plants. Glycogen is synthesized and stored mainly in the liver and the muscles. Structurally, glycogen is very similar to amylopectin with alpha-acetyl linkage. However, it has even more branching and more glucose units are present than in amylopectin. Various samples of glycogen have been measured at 1700 to 6 lakh unit of glucose. The structure of glycogen consists of long polymer chains of glucose units connected by an alpha-acetyl linkage. The branches are formed by linking C1 to C6 through acetyl linkage. In glycogens, the branches occurs at interval of 8 to 10 glucose units, while in amylopectin, the branches are separated by 12 to 20 glucose units. Structure of glycogens, which is represented here. There is a straight chain polymer, whereas some of the branched chain available at 1,6 linkage. The straight chain, which is alpha-1,4 linkage, whereas the branched chain is alpha-1,6 linkage, will produce this kind of glycogens. Cellulose is a homopolysaccharide, that means hydrolysis you will get all or single unit of monomers. Cellulose is found in plant as microfibrils. This forms a structurally strong framework in the cell walls. Cellulose is mostly prepared from wood pulp. Cellulose is a linear polymer, D-glucopyranose units. Fully equatorial conformation of beta-linked glycopyranose residues stabilizes the chair structure, minimizing its flexibility. Cellulose has many uses as an anti-cake agent, emulsifier, stabilizer, dispersing agent, thickener, and gelling agent, but these are generally subsidiary to its most important use of holding on to water. Water cannot penetrate crystalline cellulose, but dry amorphous cellulose absorb water, become soft and flexible. Purified cellulose is used as the base material for a number of water-soluble derivatives. Example, methyl cellulose, carbomethyl cellulose. The cellulose structure shown in the picture, which is a beta link between one sugar and other sugar via 1,4 linkage, which will produce cellulose carbohydrate. Chitin is another class of homopolysaccharides. It's a polymer that can be found in anything from the cells of beetles to webs of spiders. It is present all around us in plant and animal creatures. It is sometimes considered to be a spin-off or cellulose because the two are very molecularly similar. Cellulose contains a hydroxy group and a chitin containing acetamides. Chitin is unusual because it is a natural polymer or combination of elements that exist naturally on Earth. Usually polymers are made grabs, beetles, worms and mushrooms contain large amount of chitin. Chitin is a very firm material and it helps protect and insect against harm and pressures. Structure of the chitin molecule shown: two of the N-acetyl glucosamine units that repeat to form long chains in beta 1,4 linkage. So that is the responsible for this linkage. Chitosan, a spin-off chitin that has been discovered by the market, is chitosan. This is a man-made molecule that is often used to dye shirts and jeans in the clothing industry. Chitosan can be used within the human body to regulate diet programs, and researchers are looking into ways in which it can cure disease. Chitin, the polysaccharides polymer from which chitosan is derived, is a cellulose-like polymer consisting mainly of unbranched chains of N-acetyl D-glucosamines. Deacetylated chitin or chitosans is comprised of chains of D-glucosamines. When indigested, chitosans can be considered a dietary fiber. Chemical structure of chitosans: the one unit of glucose unit linked with the other unit via beta 1,4 linkage. The second group having free amino group, which is a repeat unit, will produce the polymer chain. Heteropolysaccharides, that means while hydrolysis you will get two different type of monomer sugar, which is called heteropolysaccharides, are high molecular weight carbohydrate polymers, more than one kind of monosaccharide available. Important examples includes glycosaminoglycan, GAG, the principal component of proteoglycans, and murin, a major component of bacterial cell walls. Glycosyl aminoglycan forming the proteoglycan are the most abundant heteropolysaccharides in the body. They are long unbranched molecules containing a repeating disaccharide unit. Usually one sugar is the uronic acid and the other is either galnac or galnac, glunac, or both the sugars contain sulfate groups. GAGs are highly negatively charged, what is essentially for their function. Hyaluronic acids, which is occurs synovial flood, ECM of loose connective tissues. Hyaluronic acid is unique among the CAG because it doesn't contain any sulfate and it is not found covalently attached to proteins. It forms non-covalently linked complexes with the proteoglycans in the ECM. Hyaluronic acid polymers are very large and can displace a large volume of water. A structure of hyaluronic acids given: two molecules of glucose. The first one is having beta linkage with other one via 1,4 beta linkage, which is having acid group at C5 position, whereas the second unit having second position N-acetyl amino group, which is present on the C2 position. Again gives the dermatan sulfate, which occurs in skin, blood vessels, heart vessels, etc. And the other unit having the first unit linked with the other unit via beta 1,4 glycosidic linkage. The fifth carbon having acid groups, whereas the second unit having second group, second position is N-acetyl glycosamine, whereas fourth position, which is sulfonic acid, is also present in the molecules. Chondroitin sulfate, occurrence which is occurred in cartilage, bone, heart valves. It is the most abundant. GAG means D-glucuronate, galnac sulfate. Again heparin and heparin sulfate, which is a very essential aminoglycan, which is occurrence is a component of intracellular granules of mast cells lining the arteries of lungs, liver and skin. Heparin sulfate, basement membranes, component of cell surface. Again heparin and heparin sulfate, which is a very essential aminoglycan, which is occurrence is a component of intracellular granules of mast cells lining the arteries of lungs, liver and skin. Heparin sulfate, basement membranes, component of cell surface. Murin, peptidoglycan. Again peptidoglycan, also known as murin, is a polymer consisting of sugar and amino acids that forms a mesh-like layer outside the plasma membrane of eubacteria. The sugar component consists of alternating residues of beta-1,4 linked N-acetylglucosamine. Attached to N-acetyl murimic acid is peptide chain 3 of 5 amino acids. The peptide chain can be cross-linked to the peptide chains of another strand, forming 3D mesh-like layer. Some archaea have similar layer of pseudoglycanidine. Peptidoglycan serves as a structural role in the bacteria cell wall, giving the wall shape and structural strength as well as counteracting the osmotic pressure of the cytoplasm. Peptidoglycan is also involved in binary fission during bacterial cell reproduction. The peptidoglycan layer is substantially thicker in gram-positive bacteria than gram-negative bacteria, and peptidoglycan forms around 90% of the dry weight of gram-positive bacteria but only 10% of gram-negative, and gram-positive strain is important attachment role and story-typing purpose. Again glycoconjugates, they are compounds that result from covalent linkage of carbohydrate molecules to both protein and lipid, that is called glycoconjugates. They have profound effects on function of individual cell as well as cell-cell interactions of multicellular organism. Two classes of carbohydrate protein conjugates, glycoproteins and proteoglycans. The glycolipids are found predominantly on the outer surface of plasma membrane. Glycoproteins, there are two types. One is N-linked glycosaccharides or O-linked glycosaccharides. The N-linked oligosaccharides have minimum of 5 sugar residues. N-linked attached to a polypeptide by an N-glycosidic bond with a chain amide group of amino acids and asparagines. O-linked oligosaccharides are generally short 1 to 4 sugar residues. O-linked are attached to polypeptides by the side chains hydroxyl group of amino acid serine or threonine. Polypeptide chains are hydroxyl groups of membrane lipids. Glycosylations. Glycosylation is the process or result of addition of saccharides to protein and lipids. The process plays an important role in the synthesis of membrane and secreted proteins. Majority of proteins synthesized in the rough ER undergo glycosylations. It is an enzyme-directed site-specific process. Two types of glycosylations exist. One is N-linked glycosylations to the amide nitrogen of asparagine side chains and O-linked glycosylation to the hydroxyoxygen of serine or threonine side chains. Glycosylations may play a role in cell-cell adhesions, mechanism employed by cells of immune system as well. Glycoproteins functions. Types of glycoproteins: asparagines linked carbohydrates, mucin type carbohydrates. Glycophorins, membrane proteins, source of human RBC, carbohydrate 60%. Potato lectins, the carbohydrate proteins is 50%. Function: many glycoproteins have structural functions, constituent of cell wall, form connective tissues such as collagens found in gastrointestinal mucus secretions, used as protective agents and lubricants, found in abundantly in the blood plasma. The human blood group AB and O depend on the oligosaccharide part of the glycoprotein on the surface of erythrocyte cells. The thermal monosaccharides of the glycoprotein at the non-reducing end determines the blood group. The blood groups are types and terminal sugars. A means N-acetyl-galactosamine present. In case of B, alpha-D-galactose present. In case of AB, both of them present. In case of O, neither of the above present in the molecule. In case of AB is the universal acceptor. In case of O is universal donor. Oligosaccharides are antigenic determinants. That means carbohydrates on cell surface are immunochemical markers. ABO blood groups antigens are oligosaccharides component of glycogens and glycolipids on the surface of individual cell beside blood cells. Individuals with type A cells have antigens on their cell surface and carry anti-B antibodies. Those with type B cells which bear B antigens carry anti-A antibodies. So those with type AB cells which have both A and B antigens carry neither anti-A or nor anti-B antibodies. Type O individuals whose cells bear neither antigens carry both anti-A and anti-B antibodies. Transfusion of type blood groups into type B individuals results in an antibodies-antigen reactions. This reaction clumps together the transfused erythrocytes, resulting in an often fetal blockage of blood vessels. Function of glycoproteins. Carbohydrates and proteins by themselves serve as a vast number of biological functions. Linking the two together results in a macromolecule with an extremely large number of functions. Structural glycoproteins are found throughout matrices. They act as receptors on cell surface that bring other cells and proteins together, giving strength and support to a matrix. In nerve tissues, glycoproteins are abundant in gray matter and appear to be associated with synaptosomes, axons, and microsomes. Protections. Human lacrimal gland produce glycoproteins which protects the corneal epithelium from desiccating and foreign particles. Human sweat glands secrete glycoproteins which protect the skin from the other extraordinary products that could harm the skin. Function of glycoprotein. Again, prothrombin, thrombin, and fibrinogens are all glycoproteins that play an intricate role in the blood clotting mechanism. Certain bacteria in the similar layer that surrounds outermost component of cell walls are made up of glycoproteins of high molecular weight. In addition to the forming these S layers, glycoproteins also functions as a bacteria flagella. These are made up of bundles of glycoproteins protruding from cell surface. Their rotation provides propulsions. In plants, glycoproteins have roles in cell wall formations, tissue differentiations, embryogenesis. Reproductions. Glycoproteins found on the surface of spermatozoa appears to increase sperm cells attractions for the egg by altering the electrophoretic mobility of the plasma membrane. Adhesions. Glycoprotein serves to adhere cell to cell and cells to substratum. Hormones. There are many glycoproteins that function as hormones, such as human chorionic gonadotropins, which is present in the human pregnancy urine. Another example is erythropoietin, which regulates the erythrocyte productions. Enzymes. Glycoproteins enzymes are of three types. These are all oxidoreductase, transferase, and hydrolysis. Carriers. Glycoproteins can bind to certain molecules and serve as vehicles of transport. They can bind to vitamins, hormones, cations, and other substances. Inhibitor. Many glycoproteins in blood plasma have shown antiproliferative activities. Immunologicals, the interaction blood group substance with antibodies determined by the glycoprotein on erythrocytes. Cell membrane. The cell membrane is a fluid mosaic of lipids, proteins and carbohydrates. Membrane carbohydrates are usually branched oligosaccharides with fewer than 15 sugar units. Some of these oligosaccharides are covalently bonded to lipids, forming molecules called glycolipids. Most are covalently bonded to proteins, which are thereby glycoproteins. Plants produce pectin, major component of cell wall, and the oligosaccharides on the external side of the plasma membrane vary from the species to species. The diversity of the molecules and their location on the cell surface enable oligosaccharides to function as a marker that distinguishes one cell from another. This is the function of cell membrane which we have discussed. The mosaic of cell membrane. The cell membrane contains again two types of O-linked glycopeptides and N-linked glycopeptides, which is a major role play in this cell membrane. Cell membrane matrix. The biological membrane is collage of many different proteins embedded in the fluid matrix of the lipid bilayer. The lipid bilayer is the main fabric of the membrane and its structure creates a semi-permeable membrane. The hydrophobic core embeds the transport of hydrophilic structures such as ions and polar molecules but enable hydrophobic molecules which can dissolve the membrane cross with ease. Proteins determine most of the membrane specific functions. The plasma membrane and the membrane of the various organelles each have unique collection of proteins. For example, to date more than 50 kinds of proteins have been found in the plasma membrane of red blood cells. Proteoglycans represents a special class of glycoprotein that are heavily glycosylated. These glycosyl aminoglycan chains are long, linear carbohydrate polymers that are negatively charged under physiological conditions due to the occurrence of sulfate and uronic acid groups. There are three types: keratin sulfate, heparins and heparin sulfate, chondroitin sulfate and dermatin sulfates. Proteoglycans can also be categorized by size. The large size, which is called aggregan. Aggregan is the major proteoglycan in cartilage present in many adult tissues, including blood vessels and skin. The small leucines rich repeat proteoglycans includes decorins, pyglycans, fibrinomelin, and lumicans. Synthesis of proteoglycans. The proteins component of proteoglycan is synthesized by ribosome translocated into lumen of the rough endoplasmic reticulum. Glycosylations of the proteoglycan occurs in the Golgi apparatus in multiple enzymatic steps. The sugars are added at the time of glycosyl transferase. The completed proteoglycan is then exported into secretory vessels to the extracellular matrix of the cell. Structure of proteoglycans. Again, that the carbohydrate unit which is linked via amino acids to produce proteoglycans. Some forms keratin sulfates are linked to the protein core through N-asparagine bond. Some of them form threonine or serine residues which allows the multiple CAG attachments. Function of proteoglycans. Proteoglycans are major component of animal extracellular matrix, the filler substance existing between cells in an organism. Individual function of proteoglycans can be attributed to either the protein core or the attached GSG chain. The extracellular matrix, they form large complex with other proteoglycans. There are two involved binding cations and that can affect the active and stability of proteins and the signaling of molecules within the matrix. Role of proteoglycans. As GAG, dependent function can be divided into two classes, the biophysical and biochemical. Two types of reaction it can carry out. The biophysical functions depend on the unique properties of CAGs, the ability to fill the space, bind and organize water molecule and repel negatively charged molecules. Because the high viscosity and low compressibility, they are ideal for the lubricating fluid in the joints. On the other hand, their rigidity provided a structural integrity to the cells and allowed the cells migrating due to providing the passageways to between the cells. For example, the large quantities of chondroitin sulfate and keratin sulfates found in aggregan play an important role in the hydrogen of cartilage. They give cartilage its gel-like properties and resistant to deformations. So this is the properties, and apart from that, role of proteoglycans which also do more biochemical function of CAGs are mediated by specific binding of CAGs to other macromolecules, mostly proteins. Proteoglycans participate in cell and tissues development and physiology. Hurley syndromes, the disease related with the proteoglycans metabolisms, where excessive accumulation of termatin sulfate due to the efficiency of the specific enzyme may cause enteral retardations, skeletal deformity and death in early childhood. So students, let us summarize what we have learnt in this model. Monosaccharides, the simplest carbohydrates, are classified aldose ketoses, and apart from that, cyclic hemiacetal that we learned. In case of monosaccharide derivatives, includes aldonic acids, uronic acids, and all possible classification we studied. Apart from that, disaccharides, the simplest polysaccharides occurring the hydrolysis product of large molecules, and oligosaccharides play an important role in determining protein structure that we studied in detail. Apart from that, the polysaccharides and its classification and its function we studied, especially cellulose and the storage polysaccharides and the glycosylaminoglycans, that and all we studied extensively, and also glycoproteins and glycoconjugates, proteoglycans, that also we studied. Especially in case of glycoconjugates, proteoglycans and glycoproteins play an important role in the formation of transfer of living organism that we have studied. Glycoproteins are glycosylated proteins, that N-linked and O-linked glycoproteins we studied. In case of amino acids having nitrogen which is make the bond, that will give N-linked glycoproteins, that may be probably 5 sugar unit which be present in case of N-linked glycopeptides or N-linked oligosaccharides, whereas in case of oxygen which made the link maybe 1 to 4 units of sugar unit which is present in the molecules. That and all we studied in details.